Hello everyone. Today we're continuing our deep dive of Richard Dawkins and Yan Wong's book, The Ancestor's Tale. In this episode, we're going to discuss the problem of biological continuity in taxonomy. So let's jump right in. All former videos in this series were about members of a group called the amniotes, those animals whose young develop in an amniotic sac. The last common ancestor of the amniotes lived in the middle of the Carboniferous period around 310 to 330 million years ago. Today, we are leaving the amniotes and linking up with the amphibians. Our shared common ancestor with them lived sometime in the early Carboniferous or late Devonian period between 340 to 370 million years ago. Amphibians, reptiles, birds, and mammals collectively comprise the clade Tetrapoda, meaning 4 feet, because its members ancestrally had, or still possess, 4 limbs. Snakes, glass lizards, ichthyosaurs, cetaceans, and manatees, as well as some amphibians we're about to meet, have independently lost some or all of their limbs. Amphibian means both kinds of life, referring to the fact that amphibians transition from an aquatic life to a terrestrial one as part of their life cycle. Every child learns in school about how tadpoles hatch from eggs laid in water, and those tadpoles eventually develop arms and legs before moving to life on land. But there are exceptions. Some amphibians stay aquatic their entire lives, and some skip metamorphosis and hatch from the egg as a miniature adult. Before anyone asks, no, this metamorphosis isn't recapitulating the evolution of fish to amphibians, in part because the fishy ancestors of tetrapods already had the same arm bones that we have. More on that in the future lungfish's tale. There are some 8,600 extant species of amphibians. The greater part of 7,600 of those are frogs and toads, but there isn't a real zoological distinction between these two. Basically, all toads are frogs, but not all frogs are toads. There is one family of frogs called Bufonidae, the true toads, wherein every member is known as a toad, but many frogs outside this family are often called toads too. Traits associated with these toads are relatively short legs and bumpy, leathery skin, but such traits aren't exclusive to specific clades of frogs. Thus, toads are polyphyletic. Besides the 7,600 species of frogs, amphibians also include about 800 species of salamanders and around 200 species of caecilians. Unfortunately, though, climate change, deforestation, and globalization of amphibian-specific pathogens like Batrachochytrium have resulted in the recent extinction or endangerment of many amphibian species. About 1/3 of all amphibians are threatened, while 9.2% are critically endangered. The spread of Batrachochytrium is due largely to the amphibian pet trade. The African clawed frog appears to be resistant to the fungus because both have been locked in an evolutionary arms race. However, by bringing the fungus to new lands, it has the opportunity to infect species that have never come into contact with it before. The fungus can potentially, and in many cases does actually, decimate wild populations of frogs. As for frogs themselves, they are found on every continent except Antarctica and have likely been on those continents since before they broke up. Their distribution, then, to refer back to the elephant bird's tale, reflects vicariance rather than dispersal. There are, of course, some instances of frogs being found on islands derived from continental plates, but amphibians are nearly totally lacking on oceanic islands. One notable exception is the 2 species of Fijian frog, Cornufer vitianus and Cornufer vitiensis. These frogs lay their eggs in moist soil or under rotting plants, and tiny froglets emerge from the eggs, skipping the tadpole. Direct development from froglets is actually quite common, having independently evolved multiple times. Charles Darwin marshaled the lack of amphibians on islands as evidence of evolution in deep time, because amphibians are almost always dependent on freshwater, so they can only go where freshwater exists. Since they desiccate in salt water, this places a pressure on amphibians to generally stay away from the ocean. This is evidence for a natural, not supernatural, biogeographic distribution. In one interesting case, though, the island Sao Tome has a species of frog, Tychidina newtoni, that evidently reached the island via freshwater plumes from the mouth of the Congo River. Frogs are well known for their ability to jump, and some have greatly expanded on this ability, such as Wallace's flying frog, Rhacophorus nigropalmatus, from the Malay Peninsula. Of course, frogs are ultimately descended from non-hopping ancestors, and a few have been found. One is the 10-centimeter long Triatobatrachus from the early Triassic of Madagascar. Compared to modern frogs, the front and hind limbs of Triatobatrachus were simply too short and of the wrong proportions for efficient jumping. As a 2016 paper points out, quote, jumping capacity is highly related with tibiofibula to femur ratio value. Zug 1972 reported that strong jumpers have a ratio above 1, i.e. 1.14 and 1.13 for Pseudacris crucifer and Rana clamitans respectively, whereas weak jumpers have a ratio below 1, i.e. 0.99 for Bufo terrestris. In Triatobatrachus, tibiofibula to femur ratio value is 0.66, which suggests that its jumping performance was poor. Moreover, the lack of fusion of the tibiofibula is not compatible with achieving great jumping performance. Close quote. Triatobatrachus also lacks a unique characteristic of frogs, the urostyle, which is a fusion of tailbones forming a long rod-like structure that runs parallel to the elongated iliac blades. The urostyle and the ilium together reinforce the pelvis in frogs. In Triatobatrachus, the tail vertebrae remain unfused and the pelvis is rather short compared to modern frogs. The common ancestor of frogs and salamanders lived about 272 million years ago in the late Permian. Salamanders don't jump, of course, but rather wiggle side to side like fish as they walk. Unlike frogs, salamanders retain their tail all throughout their lives, but like frogs, salamanders are typically reliant on moist environments. However, some salamanders skip the aquatic juvenile stage entirely, like Plethodon, laying their eggs under rocks or rotting logs instead. On the complete opposite side of the spectrum is the axolotl, whose totally aquatic existence will be elucidated in a future tale. Next, the common ancestor of all extant amphibians lived about 270 to 310 million years ago in the early Permian or late Carboniferous. The final extant clade of amphibians is the Sicilian, who possess no arms or legs. Not only do Sicilians not even have the vestiges of limbs, they also lack shoulder and pelvic girdles, and the ribs don't support the body. They also have small or non-existent eyes. There are some Sicilians in the fossil record that possessed small limbs and better developed eyes, such as Eosicilia from the early Jurassic of Arizona. Now that we have all the extant amphibians collected, how did amniotes, with their watertight eggs and skin, descend from ancestors who obviously had neither of those? Remember from the last tale that the common ancestor of all extant amniotes lived about 310 to 330 million years ago. Now we move further back to meet the common ancestor of both amniotes and amphibians, which lived about 340 to 370 million years ago in the early Carboniferous or late Devonian. While there are no amniotes anymore, there are amniote-like tetrapods called the reptiliomorphs, or sometimes the informal anthracosaurs. Reptiliomorpha includes amniotes and their closest non-amniote relatives like Diadectes, Siagia, Limnocoelus, and Solenodonsaurus. As it happens, some reptiliomorphs fall neatly within the 30 million year gap between our ancestor with amphibians and our ancestor with all other amniotes, precisely what you would expect under evolution. To quote paleontologist Donald Prothero's book, Evolution, What the Fossils Say and Why It Matters, quote, "We can see several trends in the evolution of anthracosaurs into amniotes. One is the fact that anthracosaurs have skulls that are very deep vertically with a narrow snout and a shortened region behind the eyes. Their skulls eventually lose the notch in the back for the eardrum. By contrast, most other primitive tetrapods, such as the temnospondyls and lepospondylus, have relatively flat skulls and bodies, with a wide snout, a large region of skull roof behind the eyes, and a well-developed notch for the eardrum. Correlated with this is evidence that limbs of the anthracosaurs were robust and held upright, so they frequently walked with their bellies off the ground. Temnospondyls and lepospondylus had a much more sprawling posture and could rarely move without dragging their bellies. The wrist and ankle bones of anthracosaurs were also modified for much more active motion than the comparable bones in other primitive tetrapods. The neck vertebrae of anthracosaurs were specialized into two bones: the atlas, which holds up the skull, as Atlas once held up the world in Greek mythology, and the second neck vertebra, the axis, the pivot joint on which the skull turns in relation to the neck. These are features found in all later amniotes and allowed anthracosaurs to swivel their heads rapidly to catch prey." Close quote. So what about the evolution of the amniotic egg? In the 1970s, paleontologist Robert L. Carroll hypothesized that the ancestors of amniotes should be no longer than about 10 centimeters from snout to cloaca, because that's about the size of extant plethodontids who, remember, lay their eggs on land. However, a 2019 study investigated snout to cloaca length in a variety of tetrapods from the late Devonian to early Permian and found no particular size trend for stem amniotes. It should be noted, however, that the fossil record of early amniotes is rather poor, but at present time, Carroll's hypothesis seems to be unsupported. On to the amniotic egg itself. A 2021 study looked at developmental and reproductive characters across vertebrata to reconstruct ancestral character states, and it found that the ancestral amniote possessed internal fertilization, copulatory organs, no larval stage, delayed oviposition, an amnion capable of myogenic contractions, a chorion, an allantois, an eggshell, gave off uric acid as excretory product, had white yolk, meroblastic cleavage, the latebra, and a cellular yolk sac. A few of those words may be unfamiliar, so we'll go through them. The chorion and allantois are parts of the amniotic egg. The former is the outermost fetal membrane, while the latter is a sac-like structure with clear fluid that helps the embryo exchange gases and liquid waste. Meroblastic cleavage is when an embryo develops a cleavage furrow, but the cleavage doesn't progress into the yolk because of how large the yolk is. This is common in insects, fish, reptiles, and birds. Lastly, the latebra is a spherical mass of white yolk within the total yolk. As for the tetrapods, the ancestral character reconstruction includes external fertilization of small eggs, holoblastic cleavage, and hatching of a larva. For reference, holoblastic cleavage is when the total embryo undergoes mitotic division. So, how did our ancestors go from the original tetrapod condition to the amniote one? Traditionally, the evolution of the amniotic egg has been seen as an adaptation to a new terrestrial environment. The inside is like a mini aquarium for the embryo, while the shell is a tough exterior for protection. However, some of these features necessarily exist together. For example, internal fertilization and the deposition of eggs with embryos that have reached the primitive streak, aka the blastopore, stage, or the early somite stage, which are paired blocks of mesoderm that form the cartilage of vertebrae and ribs, among other things, have repeatedly evolved in vertebrates. Sharks and rays, coelacanths, and other bony fish are known to possess these characteristics, which may have evolved as a response to egg predation or laying eggs in dangerous environments. Further, a large yolk is associated in multiple independent vertebrate clades with delayed egg deposition and intrauterine development or egg guarding. The amnion, chorion, and allantois are amniote inventions. However, the amnion may have facilitated the conquest of land rather than the other way around. The amnion membrane provides a physical separation from the large yolk and other extraembryonic materials, and since possessing a large yolk has evolved multiple times in aquatic vertebrates, it's entirely possible that the amnion evolved in a watery environment too. This suite of characters could have been adapted for a terrestrial environment, so all that would need to be added are the tough egg exterior and the chorion and allantois. So now we find ourselves at the common ancestor of all tetrapods. With everyone joined together, we can address the tale of the salamander. The North American salamander, Ensatina exscholtzi, is classically called a ring species because it represents a nearly continuously distributed population that circles a geographic feature, in this case, California's Central Valley, and both ends of the ring are largely unable to interbreed with each other, making them separate species. The E. exscholtzi complex originated in either southern Oregon or northern California, and then split into two lineages around the Central Valley. The inland clade can be identified by its splotched pattern, whereas the coastal clade has no such pattern. The ancestral species was Ensatina exscholtzi oregonensis, which lives from the coast of southern Canada to the Monterey Bay region. The split between the coastal and inland clades occurred somewhere in northern California, and the two clades met back up at the southern end of the valley. However, by the time the salamanders reunited at the southern end, they were too different from the original population to interbreed with it. Thus, they are essentially different species. Obviously, the Ensatina example presents a problem for taxonomy. How can E. klaberi and E. exscholtzi be members of the same species when their incidence of interbreeding is so low, way lower than what's normal for a species? Remember that the definition of the biological species concept, or BSC, is the ability to interbreed and produce fertile offspring. Clearly, that isn't happening here. The reason these two populations are considered members of the same species is that all the intermediate connections between them and their common ancestor, E. oregonensis, are still alive. Imagine if all the species connecting us and chimpanzees were still alive. Would we still say we are a different species from chimps? You might be wondering, is there a threshold of hybridization that separates two weakly interbreeding populations into separate species? Not really. One idea is to say if the probability of a reproductively viable hybrid forming is less than the mutation rate, then the population constitutes a good species. A major problem with this idea is that both the mutation and hybridization rates are often unknown for species, and those rates are going to be very difficult to acquire for many species. Even taking this idea at face value, the metric is still arbitrary. Why not double the mutation rate, or triple, or half? Why not if a single fertile hybrid is known? The point is that there are going to be subjective decisions regardless. For most organisms, there is no single mutation that suddenly results in non-viable offspring. Instead, populations fall along a continuum of breedability. Partly for this reason, researchers have erected the term speciation continuum to describe that phenomenon. On one end of the continuum, you have populations that are fully capable of interbreeding with each other. On the primrose path to speciation, the populations continually drop in the percent of interbreeding events that result in viable offspring. Whereas there was once a 100% chance of interbreeding successfully, after many generations, the chance drops to 75%. Then after some more generations, the chance drops to 50%, then 25%, then 10%, then less. Continuing this trend along the continuum, we come to the concept of the syngamion, which is defined as a group of otherwise distinct species interconnected by limited gene exchange. In other words, a group of species have a much higher chance of interbreeding with members of their own species, but can also breed with other closely related species. The ability to hybridize two or more species could even be favored under natural selection if species capable of hybridizing are more fit than totally isolated species under critical periods of environmental change. The North American oak genus Quercus famously exhibits a syngamion, although our own species does as well, since we hybridized with both Neanderthals and Denisovans. But again, what is the threshold for hybridization before two species can be considered distinct? Lastly, there isn't a point for most species where a parent gives birth to an offspring that they couldn't theoretically interbreed with. There usually isn't a clear demarcation in the speciation process. Rather, two populations typically gradually accrue distinguishing variations over many generations. Another consequence of this is that you can never abandon your ancestry. The descendants of humans will always be hominids, primates, mammals, synapsids, amniotes, tetrapods, vertebrates, chordates, deuterostomes, animals, and eukaryotes. We discussed this at length in our video Do Dogs Produce Non-Dogs? So that's the salamander's tale. Species usually arise by gradual, piecemeal processes, and sometimes this process can be directly witnessed in nature, such as in ring species. So, thanks for watching, and we'll see you all next time.